Hey everyone, in this video lecture, we are talking about a process of cell division called meiosis. First, we need to review some different types of cells in humans. This does apply to other organisms, but we're really thinking about how humans and their cells are dividing. So there are two categories of cells. We have somatic cells, somatic or soma in Latin, referring to body cells. These are cells that make up tissues and organs in the body, so muscle cells or skin cells and so on. Whereas your sex cells are specific types of cells used specifically only for reproduction. And this is because they are passing DNA down to their offspring. And they have different ploidies. So somatic cells are diploid. There are 2 copies of every chromosome in these types of cells, whereas your sex cells have a different ploidy. They are haploid, meaning they only have 1 set of each chromosome. They have half the amount of DNA. And because these 2 cells have different ploidies, they go through different kinds of replication to make more of these cells. So somatic cells, they're diploid. We want to make sure that when we go to make more somatic cells, that they retain that diploid number. So these cells go through cell division in order, they go through mitosis, which is their type of cell division, in order to make exact identical copies of their cells. We want to make sure that we retain that diploid number. So if a cell starts with 46 chromosomes, after mitosis, it should also have 46 chromosomes. Whereas your sex cells, they are haploid. They go through a different process in order to divide. They go through meiosis, which sounds similar to the word mitosis, but is a different type of cell division specifically to make haploid cells. So we'll talk about in this video how meiosis is able to create these haploid sex cells. At the end of meiosis, we begin with one cell. We will end with 4 haploid sex cells. And it's a similar process to mitosis. We're going to see that some of the names of the steps are similar, but what is a big difference here is we go through 2 separate rounds of division. And that is what's going to help us divide up those chromosomes and end up with only half the number of chromosomes in our ending sex cells. And meiosis is also really important for increasing genetic diversity. And we're going to see that throughout the different steps of meiosis where genetic diversity will kind of appear. So before the cell actually even goes through meiosis, it is sitting in that cell cycle phase of interphase where the DNA does replicate. So every chromosome will replicate itself and will have 2 chromatids per chromosome. Then the first round of meiosis is meiosis I. And the big thing here that's happening, we'll go through meiosis I in more detail, but the big thing that happens during meiosis I is those homologous chromosomes. The pairs of chromosomes are going to separate from one another. And we end up with 2 genetically diverse cells. And again, we'll go through some of the steps in detail and we'll see how that diversity appears. But the big thing here is we split those homologous chromosomes. So how this happens, the 4 steps of meiosis I are very similar in name to our mitosis steps. So we have prophase I, and it's similar to prophase of mitosis, where your nucleus is breaking down, those chromosomes are condensing so we can visibly see them. Then after prophase, we go to metaphase I, and just like metaphase and mitosis, those chromosomes are lining up in the middle. The big difference here between metaphase I and metaphase of regular mitosis is that those chromosomes are in those homologous pairs. So the pairs line up in the middle. We are not lining those chromosomes up single file. After metaphase I, we have anaphase I. Anaphase, think away. Those chromosomes are pulled to the opposite ends of the cells. Or of the cell, because we're not in cells yet. And then lastly, telophase I in cytokinesis is where the 2 nuclei form and the cytoplasm will divide and create 2 cells. Okay. Then after meiosis 1, the cells will now go through another round of division called meiosis 2. And this is much more similar to mitosis, where we have those sister chromatids that are separating from one another, and we end up with 4 unique haploid sex cells. So how meiosis 2 works, again, we're going to see the same names here. Prophase II is the first step, where the nuclei of each cell will break down. Those chromosomes are condensed so we can see them. Then we go into metaphase II, which looks very similar to metaphase of mitosis. We have the single chromosomes lining up single file in the middle of the cell. Then we have anaphase II, which pulls those chromatids apart. Each chromatid goes to the opposite end of the cell. And then telophase II and cytokinesis, where we have the nuclei reform and that cytoplasm divides. And because each cell divided into 2 cells, we now have a total of 4 cells. Each cell has only 1 copy of each chromosome. So that piece that I mentioned about genetic diversity. So those 4 haploid sex cells are not going to be genetically identical to one another. We have really unique gametes or sex cells here, which then can go on to create unique zygotes. So if you have 4 sex cells that are all different, and each sex cell goes on to create an offspring, this is why offspring, even that have the same 2 parents, offspring will not look identical. So brothers and sisters are not always going to look identical to one another. They might have similar features, but it's because those sex cells are genetically unique. And we'll see how that occurs in just a moment. The only exception to this is with twins. And twins they do have the exact same genetic material. So they're the exception to this rule, where the zygotes are genetically unique. So the first way we get some genetic diversity here. So genetic diversity, meaning differences in genes. Our genes are not the same in every sex cell, is due to this law of independent assortment. And this law states that when those homologous pairs line up randomly during metaphase I, so they're in their pair, one of the chromosomes of the pair was inherited from the egg, so the mother. One chromosome was inherited from the sperm, the father. It is random which side which chromosome lines up on. So for example, we're looking at a very simple cell, 2 pairs of chromosomes. Let's say that the blue came from the egg, the red came from the sperm in this potential offspring, this person cell. The blue in this case, with only having 2 pairs of chromosomes, just so happened to both line up on the left. Whereas here, the red are both on the right. So after they go through metaphase I, they then go through anaphase I, those chromosomes split. And at the end of metaphase I, before we enter metaphase II, we then have this cell here containing all the blue chromosomes, which were inherited from the egg. Whereas this cell over here inherited all the red chromosomes, which came from the sperm. And so then this cell will then continue through meiosis and create these haploid sex cells that have only genetic material from the egg that created this organism. Whereas this sex cells only have the red chromosomes that were inherited from the sperm. So if this particular sex cell goes on to create its own offspring, it's going to have mostly traits that were associated with the traits of the egg. Whereas if this sex cell over here were to go on and create a new offspring, it would have mostly traits of the red chromosomes or the sperm that was in this, that created this offspring, which is kind of confusing. So this is one potential option. Again, we have only 2 pairs of chromosomes, so things are pretty simple here. But we also have the option where the blue for the larger chromosome here was on the left and red was on the right. But for the smaller chromosome, red was on the left, blue was on the right. So when these chromosomes go to separate during anaphase, what we get left with is this first round of division where we have the blue big one and the red little one. Whereas over here, we have the red big one and the blue little one. So when we then finish dividing this up, we have a big blue and a little red, big blue, little red, big red, little blue, big red, little blue. Again, the color indicating in this organism where they inherited the chromosome from. And you can see that these gametes, these sex cells, are not identical to one another. The version of the trait for this R gene is the dominant version, where this person over here, I shouldn't say person, sex cell over here has the recessive gene for the R trait. So this creates mixtures of these different chromosomes. And if we have more chromosome pairs than just two, there's even more chance for differences when those chromosome pairs line up. It's completely random. So that creates different combinations of genes in the offspring, or in the sex cells that go on to create the offspring. There is also more chance for genetic diversity through a process called crossing over. It is also known as recombination. And that is because we are combining or swapping different parts of those chromosomes when we have our chromosomes line up in their homologous pairs. Basically, what happens is when those homologous pairs line up at metaphase I, they are really close together. And what can happen is the DNA of the chromosomes can kind of get caught up and wound up on one another. And this leads to entire chunks of chromosomes swapping from one chromosome to the other. And again, the colors to indicate a chromosome that was inherited from an egg and a sperm. So now you're mixing genes between the inherited DNA. And this is why, again, offspring might look like a combination of their parents. It's because the DNA is swapping between one another, and that leads to more mixing of traits. And then lastly here, this isn't so much genetic diversity, it's just errors that could occur during the process of meiosis. So nondisjunction refers to chromosomes not separating normally as they should during anaphase. And so this potentially could lead to having too many chromosomes or not enough chromosomes. So in nondisjunction, an example where this mistake could occur is in meiosis I. So those homologous pairs of chromosomes should have separated to opposite ends of the cell. Well, what happened is they kind of got stuck together in this example, and both chromosomes ended up over to the left side of the cell. So now we go through meiosis II, separate the chromatids. We end up having cells with extra chromosomes, whereas the cells that were created over here on this side have less chromosomes than they're supposed to. Over here is another type of nondisjunction, but it's happening during meiosis II. So the homologous pair is separated properly, but the chromatids did not separate properly in meiosis II. So this creates one gamete that has an extra chromosome, one gamete that has one less chromosome, and then two normal gametes that have the appropriate number of chromosomes. And so this is what leads to particular errors, or I should say diseases. And so here's two examples. We can look at a karyotype, which is like an image of the chromosomes lined up. And this can show where errors have occurred. We see that this person here has Down syndrome. They have an extra chromosome at spot 21, which is why Down syndrome is also known as trisomy. So tri meaning 3, trisomy 21, because the 21st chromosome, they have an extra. Another example of a disorder related to meiosis errors is Turner syndrome. And this is where they have one less sex chromosome. So the last pair of chromosomes are the sex chromosomes, determining if you're genetically a male or female. They only have one instead of having two.